Lettuce that tastes like meat. That's not a joke, and it's not science fiction. It actually happened. This month, scientists took a normal head of lettuce and a tobacco plant and made them grow a real animal protein inside their leaves. Not a plant version of it, not a flavor that's close enough. The actual protein. The one that gives your steak its color, its smell when it hits the pan, and that deep savory taste you can't fake with spices. Here's my promise to you. By the end of this video, you will understand exactly how they did this, why it's a genuinely big deal, why it's not as simple as it sounds, and why it's still not going to show up in your grocery store tomorrow. No hype, just what actually happened, straight from the researchers and the published study. Let's get into it. Let's start with the real problem here. If you've ever eaten a plant-based burger, you know something's missing. It looks kind of right. It smells kind of right. But something about the taste feels off, like a copy of a copy. For years, food scientists have known exactly why. It's one molecule. It's called myoglobin. Myoglobin is the protein that stores oxygen inside animal muscle. Cows have it. Pigs have it. You have it too, in your own muscles. It's actually the same protein your body uses to help oxygen diffuse into working muscle tissue, which is part of why some animals that dive or hold their breath for long periods, like whales, have absurdly high concentrations of it. Cats even rely on a version of it as a source of taurine, an amino acid they can't make enough of on their own. But here's the part that matters for food. Myoglobin holds a tiny iron core called heme, and heme is basically the flavor engine of meat. Heme is why raw meat is red. It's why meat turns brown when you cook it. And when heat hits it, heme kicks off a chemical reaction called the Maillard reaction. That's the exact reaction that creates the savory, almost metallic taste we associate with real meat. Take heme out of the picture, and you're left with something that just tastes like seasoned protein. Not meat. So for years, companies have tried to solve this. Impossible Foods, for example, makes their burgers bleed using a similar heme-containing protein called soy leg hemoglobin. But they grow it in genetically engineered yeast, in giant industrial fermentation tanks. It works. But it's expensive. It needs sterile factories, huge energy input, and specialized equipment. That approach even has a name in the industry: precision fermentation. It's powerful, but it's basically brewing beer at pharmaceutical-grade precision, and that infrastructure isn't cheap to build or run. So here's the question researchers at Imperial College London started asking. What if you didn't need a factory at all? What if a plant, a normal, growing, photosynthesizing plant, could make this protein itself? That question is exactly what led to the study we're talking about today. This research was published on August 6th in the journal Frontiers in Plant Science, led by Dr. Alexia Groff at Imperial College London, working with the university's Bezos Center for Sustainable Protein and a Cambridge biotech company called Kiome, along with researchers from the University of California and Nanchang University. It's actually the first published report of any higher plant stably producing myoglobin at all. Earlier attempts had only managed short-term, transient expression, not the real, inheritable thing. Their goal was simple to say and very hard to do. Get lettuce and tobacco plants to produce myoglobin, the actual animal protein, cloned from pig and cattle genes. Now, stop for a second. Tobacco and lettuce making an animal protein. That sounds almost impossible. So how do you even do that? Let's break it down step by step. Every plant cell has a nucleus. That's the main control center, where most of a plant's genes live. You'd think that's where you'd insert a new gene, but the researchers didn't go there. Instead, they targeted something smaller, the chloroplast. That's the part of a plant cell responsible for photosynthesis, the process that turns sunlight into energy. Here's the twist that makes this whole study clever. Chloroplasts weren't always part of plant cells. Billions of years ago, according to accepted scientific theory, they were free-living bacteria. At some point, an ancient cell swallowed one of these bacteria, and instead of digesting it, kept it around permanently. Over time, that bacterium became the chloroplast. Because of that bacterial past, chloroplasts still carry their own tiny separate genome, completely apart from the plant's main DNA. And that turns out to be the perfect place to insert a new gene. 2 reasons. First, numbers. A single plant cell contains many chloroplasts, and each chloroplast contains multiple copies of its own tiny genome. So when researchers insert one gene into the chloroplast, that plant cell ends up with thousands of copies of that gene. Thousands. That's why chloroplasts can pump out far more of a target protein than inserting the gene into the plant's main nucleus. And the researchers actually tested this directly. When they compared chloroplast insertion against inserting the same myoglobin gene into the plant's nuclear genome, the chloroplast version produced roughly 3 times more protein. Second reason, location. The chloroplast is also where plants naturally build something called heme, the same iron-based molecule found in myoglobin. Plants don't use heme for oxygen storage like animals do. They use it as a building block for chlorophyll, the pigment that makes leaves green. But it's the same core molecule. So by putting the myoglobin gene right there, in the chloroplast, the protein gets built in the exact same neighborhood where its essential ingredient, heme, is already being made. Quick detour, because I think this matters for context. Using plants as living factories for animal or human proteins isn't actually a brand new idea. It's called molecular farming, and it's been a real field for decades. Plants have already been engineered to produce human insulin, antibodies, and even vaccine components. A tobacco-produced antibody for treating HIV and an insulin grown in safflower plants have both cleared regulatory approval in the European Union. And a plant-made enzyme therapy for a rare genetic disorder was approved for patients in the United States back in 2012. So the idea of growing a complex biological protein inside a crop, instead of a steel fermentation tank, already has a track record. What's new here isn't the concept, it's the target. Nobody had gotten a plant to stably produce myoglobin for food before. Here's where it gets a little wild. Plant cells are wrapped in a tough wall made of cellulose. You can't just inject DNA through that like you would with an animal cell. So the researchers used something called a gene gun. Yes, it's literally called that. It's a device that coats microscopic particles of gold or tungsten with copies of the myoglobin gene, then fires them under high pressure directly into plant seedlings. The particles physically punch through the cell wall without destroying the cell, dropping the DNA right where it needs to go. Before any of that firing happened, though, there was a lot of lab work behind the scenes. The researchers first cloned the actual genes for pig and cattle myoglobin, then rewrote, or codon-optimized, those gene sequences so a plant's cellular machinery could read and build them efficiently, since plants and animals don't always use genetic code the same way. They assembled the finished gene constructs using a cloning method called Golden Gate assembly, essentially a precise way of snapping DNA building blocks together like Lego pieces, before those constructs ever went anywhere near the gene gun. After firing, the plants were grown out in controlled lab conditions, tested to confirm the gene had actually settled into the chloroplast genome, and then grown all the way to full flowering adult plants. And here's the part that really shows this wasn't a one-off fluke. Those plants passed the myoglobin gene down to their seeds. Their offspring inherited it too. The team didn't stop at tobacco and lettuce either. They also tried inserting a version of the gene, this time for cattle myoglobin, into the chloroplasts of a green algae species called Chlamydomonas reinhardtii, which scientists often use as a simple test organism. It worked, but nowhere near as well. The algae produced myoglobin at less than 0.25% of its total soluble protein, compared to roughly 2.7% in tobacco and 1.5% in lettuce. In other words, leafy land plants turned out to be dramatically better hosts for this than algae. Okay, so the gene went in, but did the plants actually make working myoglobin? This is the part that matters most. Yes, the plants did produce it. Using lab techniques like mass spectrometry and antibody-based detection, Researchers confirmed the tobacco and lettuce leaves were making real myoglobin protein, and that it folded into the correct shape, the same structure you'd find in an actual cow or pig. Not a distorted version, the real folded structure. And get this, the plants seemed completely fine doing it. No stunted growth, no visible stress. They looked and grew just like normal, unmodified lettuce and tobacco. The researchers even specifically checked for a subtler kind of damage, oxidative stress, which can happen when a cell's machinery gets overloaded, and reported finding little evidence of it in the modified tobacco. That matters, because it suggests the plants can carry this extra manufacturing job without quietly wrecking their own cells in the process. Now let's talk yield, because this is where scale comes in. The tobacco plants produced myoglobin at roughly 800 milligrams per kilogram of dried leaf. The lettuce came in almost identical, around 810 milligrams per kilogram, dried. For comparison, actual beef muscle contains somewhere between about 8,000 and 11,000 milligrams of myoglobin per kilogram, dry weight. So yes, gram for gram, the plant version is around 10 times lower than real beef. But here's why that's not a deal breaker. A field of lettuce or tobacco can be grown fast, at massive scale, on far less land and water than raising cattle. Even at a lower concentration per plant, researchers argue the total output per acre could end up competitive with traditional livestock farming, once this is scaled up. Now, I promised you no hype. So let's talk about the actual limitation here, because there is one, and the researchers were upfront about it. Myoglobin only works, meaning it only has color, flavor, and function, if it's holding on to that iron-rich heme core. It's not enough to build the protein. You need the heme to actually click into place inside it. And here's the problem. When researchers checked the plant-grown myoglobin, only about 35% of it had heme properly attached. Compare that to precision fermentation in bacteria, like the microbial method companies such as Impossible Foods use, which reaches around 80%. So more than half of the plant-made myoglobin, right now, is technically present, but functionally empty. No color, no flavor payoff. Why does this happen? Because plants only make just enough heme for their own normal needs, mostly for building chlorophyll. Suddenly asking the plant to fill thousands of new myoglobin molecules with heme overwhelms that supply. The plant did ramp up heme production a little in response. Researchers found elevated heme levels in the modified plants, but not nearly enough to keep up. The researchers are clear about what comes next. Future versions of this technology will need to also engineer the plant's heme-making pathway itself, not just the myoglobin gene, to fully close that gap. Now here's something important for context. This lettuce and tobacco study isn't happening in isolation. There's another major player working on a similar idea, using a completely different plant strategy. A company called Moolec Science has been doing something similar, but with soybeans instead of leafy greens. Their product is literally nicknamed Piggy Sue. Back in 2023, Moolec reported that their engineered soybeans were producing pig protein at over 26% of the plant's total soluble protein, so much that the beans actually turn a visible pink color. So why grow this in leaves at all, if soybeans already work? Because leaves and seeds each come with their own trade-off. Leafy crops like lettuce and tobacco grow fast and produce a lot of biomass per acre. That's their strength. But leaves are also mostly water, and they start breaking down almost immediately after harvest, which makes storage and transport genuinely difficult. Seeds, on the other hand, like soybeans, naturally dry out and can sit in a grain silo for months without spoiling. That makes them much easier to harvest, store, and process using farming infrastructure that already exists worldwide. So really, these aren't competing technologies. They're two different tools: fast-growing, high-biomass leafy crops, and stable, easy-to-store seed crops. Both are chasing the exact same goal: making animal proteins without the animal. Let's zoom out for a second, because the why here is bigger than just better-tasting burgers. Raising livestock takes a huge amount of land to grow animal feed. huge amounts of fresh water, and it's a major source of methane and nitrous oxide, two powerful greenhouse gases. There are also long-standing animal welfare concerns tied to industrial-scale livestock farming that researchers in this space point to directly. The global market for meat alternatives is already worth somewhere between roughly 6.7 and 8.1 billion euros a year, and it's projected to keep growing between about 8 and 12% annually over the next decade. Plants, on the other hand, are what scientists call primary producers. They build their own biomass directly from sunlight and carbon dioxide. No feeding, no years of raising an animal before you get anything usable. That efficiency gap is exactly why researchers are so motivated to make this leaf and seed approach actually work at scale. As the director of Imperial's Bezos Center for Sustainable Protein put it, this points toward using plants as scalable, low-input production platforms that could work alongside microbial fermentation, not necessarily replace it, diversifying how and where these ingredients get made in the first place. Good question. Let's talk regulation, because this is where a lot of promising lab science quietly dies. In the United States, genetically modified crops go through review by the USDA and the FDA. And interestingly, Moolec's pig protein soybeans already cleared a USDA review process, confirming they don't pose a greater risk than regular, unmodified soybeans. That clearance is now letting them run open field trials in states like Ohio, Missouri, and Iowa. Here's an interesting safety detail specific to the lettuce and tobacco study. Because the new gene was inserted into the chloroplast, not the plant's main nuclear DNA, it can't spread through pollen in most flowering plants. Chloroplasts are passed down only through the mother plant, seed to seed. That naturally limits the risk of this gene spreading uncontrolled to nearby wild plants or other crops, which is a real concern regulators in places like the European Union take seriously. And as I mentioned earlier, this isn't regulators' first time evaluating a plant engineered to make an animal or human protein. Molecular farming for medicine has already gone through this process successfully for other products. That doesn't guarantee a fast path for a food product, since food and drug approval pathways are different, but it does mean the regulatory questions here aren't unprecedented. Let's be honest with each other here. Not soon. This research is what scientists call proof of concept. That means yes, it works, yes, it's real, and yes, it's published and peer-reviewed. But it's still early-stage lab science. There's no product on a shelf. There's no approved food ingredient yet. Experts covering this story have been clear that turning this into an actual commercial product is likely still years away. What needs to happen next? Researchers need to boost that heme attachment rate. They need to figure out affordable ways to extract and purify the protein from plant leaves at industrial scale, which is its own expensive challenge. And then, this needs to clear food safety regulators in every country where it would be sold. Interestingly, meat flavor might not even be the first commercial use for this. Kyome's chief scientific officer, one of the study's co-authors, has floated a different, arguably simpler near-term application. An edible lettuce engineered to express myoglobin could potentially serve as an iron-enriched, biofortified food in its own right. Heme iron is far easier for the human body to absorb than the plant-based iron you'd normally get from a salad, depending on how legislation around it eventually shakes out. That's a lower bar to clear than tastes exactly like beef, and it could realistically arrive first. So let's bring this all the way back to that promise I made at the start. Scientists really did get lettuce and tobacco to grow real animal myoglobin inside their chloroplasts, using a gene gun and some very clever biology. It folds correctly. It's inherited by future generations of the plant. It builds on a decades-old field of using plants as biological factories that has already put other engineered proteins through regulatory approval, and it points toward a future where the flavor of meat doesn't have to come from an animal at all. It's not perfect yet. The heme problem is real. The timeline to your dinner plate is long, but this is a genuine, peer-reviewed step toward solving the single hardest problem in plant-based meat, making it actually taste like meat. If you want more science stories broken down like this, no hype, just what the research actually says, hit subscribe. It genuinely helps this channel grow. Drop a comment and tell me, would you eat a burger made with plant-grown myoglobin? And if this video actually helped you understand something you'd seen in the headlines, send it to one person who'd find it interesting. I'll see you in the next one.